What's that? I'm not surprised. I mean, so I'm not saying.
So it makes sense that if we make here's what we're three, they would turn the ACF computers and then they're not, if they got here.
I'm so stressed at this point. Yeah. What's that? We started there. It's okay. What? Sure. Okay, are these? Yeah. So easy. Oh my God. Yeah. Yeah. It's really exciting. It's not really exciting.
I'm running and you got two people watching. It's a problem. Ari definitely because he I was trying to get it up. Probably your mom.
Okay.
So you're probably like your house.
Hello, everybody watching. I don't know if they can.
I doubt they use the chat. So.
Okay.
I'll adjust the camera once you get started. Good luck.
Oh, yeah. We're sure one thing. Oh, yeah. Go back, talk amongst yourselves. Maybe he's like me. He should make it.
I appreciate my slide.
Yeah, I got him like free food. No, I didn't jump in. I I should have visited a restaurant, but NPS people. Right, because there are no real labs here. It's all like, it's all administrative. Right.
Yeah, starting to get up.
That's fine. I don't have to hear you.
That's true, you're not evaluating.
He's been around long weekends.
All right. Hey guys, so I'm really pleased today to introduce Rebecca to present her thesis work. Those you know me know I get really nervous about introductions, so I tend to type them up verbatim. I don't know why, especially for a student who at the university for 4 years and also simultaneously was really productive in a couple of human genetics laboratories, primarily with Carol Ober doing human Hutterite genetics, and she ended up on 3 papers during that time, plus from one from her postbac at University of Michigan. So she did really great in the interviews. I remember from her because we bonded over war stories of trying to optimize allele-specific PCRs, and she was recruited heavily by the program here in HSG. And I was surprised when she came to rotate in my lab later because I was just sort of just getting started and didn't have that much of a human genetics footprint. For her rotation, she wanted to try something totally different, and so we came up with something, and we called it. It's pretty ambitious because you want to try something different from experience. We called it the Crapture experiment, which was we were trying to capture all the crap in the genome and sequence it. They just discovered triplet expansions caused a variety of neurological diseases they hadn't known about, like C9orf for ALS. We wanted to try and find more of these things. We thought we'd combine like an exome capture for highly repetitive sequences with this brand new long read sequencing called PyBio. We thought this would make a great rotation project. It failed epically. And it turned out that whole Crapture thing, not only that it's a crappy part of the genome, but if anybody ever did any PacBio sequencing in the early days, like a 20 to 40% error rate, it turned out we were really shooting ourselves in both feet at once. So she did great, but I was surprised she wanted to come back to the lab after that experience. But nonetheless, a year later, she not only came back, but she decided she wanted to try something crazy again. And that was to try and develop a new method for looking at RNA and synapses. So I don't know, once bitten, twice shy, didn't apply in your case. Anyway, I don't want to tell you about that story because that's her story and she's going to tell you about it. But I will tell you about it, even beyond that great work that she's going to tell you about. She made a lot of contributions to the lab during her time here. And it really kind of represented her ability to make lemon out of lemonade at times. Or sorry, the other way around, lemonade out of lemons. Because at graduate school, things sometimes work and sometimes don't. And she ended up with 3 additional papers out of my lab by taking things that almost didn't work in one way and finding a good use for them somewhere else. So she made key contributions to collaborators working with characterizing what human motor neuron profiles are out of control samples from another experiment. She helped me correct a statistical error in the field that was having everybody think every psychiatric disease was caused by Fragile X mental innervation protein, when it was really just that those are all long genes. And she really helped out with this third project where we were with collaborators, we were struggling over understanding weird gene expression phenotype in the CNH3 mutants. And she did some key experiments that really unjammed a project for that other lab where her data came out, again, very strange looking, but I trusted her hands enough at that point that we had to come up with an explanation that fit the data. And that explanation turned out to be what's true with those mice, and that kind of fixed the project that was off the rails. So she's made a lot of contributions to the lab beyond the great work that she's going to talk about. But because we could trust her work so much in that area that she was able to make those gains. All these examples speak to her ability to kind of march on and generate useful results in trying times. I think it's also notable she was able to accomplish a lot of her thesis work during a period in her life where she was supporting her friends and family through a lot of personal health tragedies over the last couple of years. I know that not everybody who wanted to be here is able to make it today, but I'm sure wherever they are, they're as proud of you right now as I am for all the work that you've done.
Live stream.
Yeah. Oh, the live stream. Okay, hello. You're as proud as I am of her. She's done fantastic work in the lab. I'm looking forward to hearing what she's going to present her final work for us now. And please join me in welcoming Rebecca in the presentation of her thesis work.
Rebecca Alniga, and as Joe said, I'm going to present today the regulation of local translation in neural processes of very specific cell types. So in basic biology courses, we're taught the very dogma of the cell, that DNA makes RNA, makes protein. And it's always shown with a diagram like this, a very spherical cell with the nucleus in the center harboring the DNA and the translational machinery outside of it. But in the brain, you have cells that are far from spherical. You have the cell body, but also these long axons and dendrites that extend far away from the cell body. So proteins are still needed even at the tips of these axons and dendrites. How do they get there? Some of these proteins get there through local translation, where RNA is transported down axons and dendrites and translated locally. This local translation is what underlies my talk today. I hope to first give you a little bit of background of how did we first observe local translation, then a review of previous methods that were used to study it, then a novel method that I created, SnapTrap, to study it, and what we found by utilizing it on 2 different trap lines. Then finally, a little biological relevance of a couple of the local translation candidates that I found through this work. But first, a little background. The first evidence for local translation was found through electron microscopy when both ER and ribosomes were observed in neurites. You can see here on this electron micrograph polyribosomes at the base of a synapse and a dendrite. This local machinery was determined to be functional when radiolabeled leucine incorporation produced radiolabeled protein in neurites that were detached from the cell bodies. This experiment showed that the local translation machinery was able to make local protein. Besides the machinery, you also require RNA. So it was in the 1980s that Lawrence and Singer observed this gradient of mRNA localization instead of just random diffusion throughout the cell. And this organization was driven through an RNA sequence called an RNA zip code. Since then, multiple zip codes were determined to be binding motifs for proteins like FMRP. These binding proteins bind the RNA and deliver them to subcompartments within the cell. For example, this FMRP binds RNAs and shuttles them to synapses. Then in the 1990s, local translation was determined to have a function in LTP, long-term potentiation. LTP is the strengthening of synapses that aids in learning and memory. When a synapse is sufficiently stimulated, you can see the synapse strengthening in intact neurons. This also happens in synapses where the neurite is cut away from the cell body. Only after translational inhibitors are added does this phenomenon go away. So this shows that the local translation is needed for LTP development. But it wasn't until the 2000s that people wanted to look at a more high throughput method of which RNAs actually make it out to neurites. Looking at these 3 studies, we can see the inconsistencies and discrepancies between these studies. These 3 found absolutely 0 transcripts in common for their candidates. It's unclear if this is due to biology or simply due to the differing methods that they used to identify them. But the question remains, which RNAs are localized to neurites? Previous studies tried to answer this first by using in vitro methods, where neurons are grown on a fine mesh that cell projections can grow through the pores. This allows for a very clean separation of cell processes from their cell bodies. However, it's a loss of the neuronal environment, so it's really rather inconsistent with in vivo data. In vivo studies, on the other hand, mainly utilized places like the hippocampus, where neurons grow in a laminar projection, where you can cut away the cell bodies from their neurites. This, however, only works in a limited number of cell types in the body, and it's highly contaminated with glial cells in that layer. So all of these cons led me to create a novel method called snap trap. For this method, I used a discontinuous gradient to harvest neurites. This harvests synaptoneurosomes, which is a presynaptic and postsynaptic baton in the surrounding glial cells. The advantage of this method over the others is that it can be done in vivo and from a greater number of cell types. However, the fraction still contains glial contamination. So I decided to enrich for neuronal RNA using TRAP, or translating ribosome affinity purification. This method utilizes a GFP-tagged ribosomes under a cell-specific promoter. This can be done in vivo, and you can see here the SNAP25 line is a pan-neuronal line, which expresses the tagged ribosome only in neurons. The RNA on these ribosomes can then be aminoprecipitated to create a cell-specific sample. So combining these two methods, I created SnapTrap. This creates 4 samples. Please note the acronyms as I'll be using them throughout the entire talk. The first sample is the whole cell homogenate. This represents the whole brain RNA. The second is TRAP on the whole brain homogenate, which represents neuronal RNA of the brain. Then, from the synaptic fraction, I harvest 2 additional samples. The first, just the fraction itself, which represents RNA from both neuronal and glial, so a mix of cell types.